His last book, Reversing Human Aging, reviews the recent research into telomeres and identifies a mechanism for the regulation of aging. Now, he is currently compiling a new magnum opus which has more than 3,500 clinical references. I understand it will be entitled Cells, Aging, and Human Disease. Here to tell us about the future possibilities, please welcome back for the last time, Dr. Michael Fossel.
If it's the last time, does that mean I'm not coming next year? It's the last lecture, and in some ways it's probably the most radical lecture that we'll have had. This is for you, Lady Olga Maitland. Right, we're both gardeners. I promised I'd put that back up for her since it's my garden. The peonies you can see, but sorry about the lupin. So, we're going to talk about, if I can get my computer to recognize me again, aging, which we've been talking about for 2 days. The difference is that I'm going to talk about reversing human aging. Let me put this in context, sort of historically. If you look back over the last couple of hundred years, this is the lifespan 200 years ago and 100 years ago currently, and this is the estimate of what we could do. We could rectangularize it or compress that morbidity. However, during that threefold increase in lifespan, at no time have we had any impact on that estimated 120-year limit. And that's what I'm talking about. A very different question, can we affect that? The answer is probably. That's what we're looking at. My interest is clinical. I mean, I have no interest in whether telomeres cause aging, and I never said they did, or anything else causes aging. My interest is where can we most efficiently and effectively intervene in the diseases that we get with aging. Having said that, let me bring you through some of the theoretical model. Luckily, Marios Karyazis has already told you all about it, including the shoestring, so I don't have to do that again. Age-related disease really is the result of old tissues, which in turn are the result of senescent cells. If we look at what goes on within a cell, there are a lot of things that happen. One of the earliest is that nuclear gene expression changes. The result of that is that there's a change in protein turnover, as Dr. Naji already told you about. As it turns out, if you look at the mathematics of that, it becomes a critical portion of intracellular aging. But there are also marked changes not only within the mitochondria, but in the entire mechanism of how we deal with free radical metabolism. Not only do you find that as the cells get older, there's an increase in the ratio of reactive oxygen species per mole of ATP, but you also find that we're no longer as good at maintaining those reactive oxygen species within the nucleus. Remember again that eukaryotic organisms are the only ones that separate the DNA from the mitochondria, and as we get older we begin to fail at doing that. Furthermore, we're less effective at trapping the escapees, the reactive oxygen species that make it out. Finally, we're less good at repairing. We still repair DNA, but the effectiveness of that repair goes down. DNA is the only molecule in your cell that you repair. Everything else you turn over, which is why that turnover becomes so critical. But the important feature is not just what happens within cells, but what happens between cells. You know, no cell is an island, each is a part of the continent. So while within cells we have a number of changes that occur, which I won't go into in much detail, more importantly perhaps are what goes on between cells. We have perfectly innocent bystander cells, end organs for example, that are affected by cells that are aging. These are the kind of differences you're looking at. We have differences within the space between cells. We have cell-cell dependencies. For example, your heart depends on the vessels. Doesn't matter whether your heart ages or not, the vessels certainly do. We have inflammation, cytokine changes, and we have what might be loosely called hormonal changes, including local hormones like trophic factors. If we take an overall model of this, and then it's going to look a little complex, but we'll go on to the clinical data in a minute. If we look at all the accumulated cell dysfunction with age, one example of this would be free radical damage. And let's see how that happens. We've mentioned some 4 factors already, but if we go upstream from that and say what caused those, we find there are changes within the mitochondria, which is dependent on the nuclear DNA. There are changes in membrane repair, again dependent on the DNA. Changes in protein production, predominantly turnover, and changes in protein turnover directly having an impact on DNA repair. This is something you'll be glad to know I'm not going to get into. You can if you want to. It explains exactly why it is that as protein turns over less well as we get older, you end up with more damaged molecules anytime you go looking for a useful molecule within your cell. But in any case, all of these factors are affected by altered patterns of gene expression, and another name for that simply is cell senescence, which occurs because cells divide. So let's get rid of the bottom and start again. Cells senesce, and we've got 4 types that I'm just looking at here: vascular endothelial cells, microglia, chondrocytes, lymphocytes. The first 2 are both cells that have an impact on other cells. That is, we start with vascular endothelial cells, for example, and they affect non-dividing cells, like myocardial cells. Likewise, microglia have effects on neurons. So we get myocardial infarctions, we get Alzheimer's dementia, we get osteoarthritis, and we get immunosenescence. These are just 4 examples of this. This is how it happens. This brings us back to you, Marios. Long telomere cell, and note the yellow thing. This is sort of the cartoon version of the heterochromatin sheath that surrounds the end of the telomere and has an impact on how gene expression changes. You might say that the critical factor as we age is the tune we play. You know, if I go to a symphony tonight and I listen to Mozart, and tomorrow night I go and I hear John Cage, the difference is not that they fired the first violin. The difference is not that the oboe's out of tune or the piano's out of tune, or that's not the difference between Mozart and Cage. The difference is the score. And all of your genes are intact as your cells get older. The problem is they're playing a different score. And one of the things that modulates the score within cells is this heterochromatin sheath. So as the cell ages, you get this changing in the telomere length, the sheath, and the effect is an altered pattern of gene expression. The upper right is the cartoon version of gene array, and as we get older again, you have an altered pattern of gene expression. This is one example. I'm skipping the others. I skipped the clone and I'm going to skip the knockout mice. But if you advance this too early prenatally in certain cell lines, you get the favorite children of mine. Again, only in certain cell lines. This is, again, the cartoon version. Most people would say this is what happens with cells. Germ cells maintain their telomeres, did I say telomeres? I'm sorry. Telomeres indefinitely. Remember that if you look at the germ cell and you go back retrospectively, I mean, all of you came from germ cells that came from germ cells that came from germ cells. In a biological but realistic sense, each of you is 3.5 billion years old. You look marvelous. You look just great. And this is despite the fact that in all that time, there's been free radical damage and there have been lipid changes. But the point is that the germ cell line is capable of maintaining homeostasis for billions of years. Not actually all that well. Most of them died. There are mutations, which is why you're not a slime mold. But within the body, most cells, I take that back, many cells, senesce. There are some stem cell lines, I mentioned yesterday, that senesce at a slower rate because they partially maintain telomere length. There are cancer cells that maintain it to our detriment. These are the Hutchinson-Gilford children. They start with shorter telomeres. And these are the Werner syndrome, the adult onset progerics. They have a faster loss, but they start with normal length. Now, this is what really happens. That's reality. Now, liver cells divide at one rate. It depends on whether you take out liver cells or drink alcohol. Skin cells another rate. Neurons very slowly, and so on. The reality is much more complex than the cartoon. Let's look at some of the diseases we associate without getting older. And here are some examples. Dermal aging. These are the sorts of things that you and I see occurring within cells right down at the submicroscopic level, the ultrastructural level. But the result of this is shortened telomeres. What we see clinically, or histologically I should say, is things we've known about for 100 years. And the outcome is what you see in the mirror every morning, which is this. If we look at a different system, immune senescence, we have the same sort of thing going on. Gradual loss of telomere length as a result of continued lifelong cell division. And the effect of that is this sort of thing. You know, your immune system is sort of like your brain as you get older. It knows a lot more, but it can't remember it very well. I like to think that people don't actually lose their memory as they get older. They just get more honest about it. But the result of that clinically is that old people, for example, die of pneumonia, and 20-year-olds don't, generally. Looking at a different system, in this case a critical one, Remember that most deaths in developed nations are a result of this process. We find that there's cell stress within the endothelial cells, things like rheologic factors, flow characteristic changes, as well as toxins, smoking, high glucose, and so forth. The result of that is cell loss, cell division, shortened telomeres, altered gene expression, and this precedes, by the way, the onset, and we see it in vivo in people, precedes the onset of what we regard as the classic model of atherosclerosis or atherogenesis, all the usual cast of characters. And the result, again, is what I see every day in my practice. This is something I've seen in a textbook, almost a quote actually. The idea is that cell senescence couldn't possibly cause heart disease because myocardial cells don't senesce. Right? Nobody said they did. Let me take out those little pink terms and substitute this. Cholesterol can't cause heart disease because myocardial cells don't accumulate cholesterol. Right, they don't. No one ever said they did. It's the coronary arteries that have all the problems. The same thing goes on, as I say, with cell senescence. Sloppy thinking. Osteoarthritis, same thing. Cell stress, changes in those cells, replacement of those cells, altered gene expression. The result is that. And the result for you and I is this sort of thing. Another system, osteoporosis. This has certainly been measured more carefully recently with regard to cell senescence, and we see there's a shift. In fact, the osteoblasts begin to divide more. They lose telomere length, gene expression changes. The result is an imbalance. In fact, you come out ahead, or the osteoclasts come out ahead. Clinical outcomes, the same thing. Finally, this system. Now, the previous systems I mentioned, we've looked at telomere loss and seen it. It precedes the disease, doesn't prove cause, but it's there every time. In this system, we have a hard time separating out microglia in living human beings. People seem not to volunteer for this. But the question is, do we find that there's cell stress and cell loss within the microglia system? Remember that most of the brain is composed of not neurons, but glial cells. Most of those glial cells don't divide, oligodendrocytes, for example. Microglia do. They're not actually an indigenous cell population, but they may as well be practically. And we know that they're implicated early on in the onset of Alzheimer's disease. But the question is, do we see telomere loss in these cells or not? And we don't know. However, if we do, it may explain, again, the sort of thing that we've known about now for decades. And the clinical outcome, of course, is dementia. Now, ask yourself an important question. Can we do anything about this? I mean, all of this is an interesting theoretical construct, but does it have any bearing on what we can do? Let me give you a sort of a spectrum. There's, can we affect aging in cells? Can we affect aging in tissues? Can we affect aging in you? And the answer is yes to the first two and not yet to the third one. And I'll show you what I mean. Most cells, fibroblasts for example, have limited number of cell divisions and they show advancing cell senescence. However, if we replace telomere length, the answer is yes. Instead of 60 division, in fact, we're well over 2,000 divisions now. And these cells, in all ways that we can measure, function normally. Cell gene expression, for example. In fact, let's look at just that. If we take young cells, they have a characteristic pattern of gene expression that's cell phenotype specific. But as we get older, if we look at those gene expression, it alters. Now, if we take telomerase and use it to re-extend telomere length, what happens? It goes right back, there's the reference for you, right back to what it was before. Very nice. But again, you and I aren't cells. Can we extend this to something more important to us clinically? Does any of this have anything to do with that? And there the answer is yes. In many ways, this is probably the most important slide, certainly in my talk. I'll give you the model. What's been done here is that we take a severe combined immune deficient mouse. This mouse rejects nothing. I could transplant my belt onto this thing and it would stay. Now we take the two major indigenous types of cells found in your skin. We take keratinocytes and fibroblasts, major two cell types. That means we're losing, depending on how you divide this, a couple of dozen other types, like melanocytes and sebaceous gland cells, and so on. But we take the two major components, and we take those cells and we put them onto a mouse under a little teeny plastic plastic envelope, and we watch those over a period of days to weeks. They layer out and they form what looks like normal human skin. That is, the layering looks good, histological levels right, the karyotype's normal, and there are important structural considerations like the interdigitations between the two layers, totally characteristic of normal young skin. They don't slough, hard to get apart, very effective. Now, if we do that same thing with senescent cells, we get this sort of thing. We get what looks like old human skin. What you find is it's thinner. There are no interdigitations. There are microbulli. Any little traction sloughs it right off. Characteristic of old skin, a model. And now, obviously, the major question, which is what happens if you take those senescent cells and re-extend the telomere? You don't do anything else. The answer is you get young skin again. Well, look at what we've done. We've taken old cells and literally reversed aging at the tissue level. Reference to that is Walter Funk's article back 2 years ago now. Well, since that time, we've gone a bit further, but not in any interesting place for you and I clinically. But still, let's look at some of that. Osteoblasts, nice balance in young cells, looked at in vivo. True, this changes, but also in vitro this changes, and you get osteoclasts predominating. If you telomerize them, you're back to a normal balance and a reference. Different model. We go to arterial cells, in this case arterial endothelial cells, human endothelial cells, functioning normally. Senescent cells, telomerize them, we get normal characteristics. These things will grow little vessels, not only if we transplant them, but they'll do it in vitro in the right conditions and a reference. So all of these have potential for human clinical intervention. So can we do it? There are two ways to do this. If right now the lights went out in this room and I wanted to turn the lights on, there are two ways we could do that. We could, not knowing where the light switch, anybody know where the light switches are? I mean, I don't. We could bring in a generator, we could bring in a large wire, we could bring in a huge light and turn it on. That's gene transfection, okay? The telomerase gene is present in every cell in your body, barring cells that don't have DNA like your erythrocytes. But it's in there. The problem is we can't find the light switch. So what we do is we take a human gene, we put it in a cell that already has the human gene. I talk about orthomolecular medicine. This is perfectly normal stuff. It's already in there. You could argue it's not even gene therapy. But the problem is it's clumsy. And the take on this at best is very poor in vitro, which is why it's done that way, because then we can select the cells we succeeded at. And in vivo, it's worse. We have to use a gene gun, for example, and fire it in under high pressure. We'd like to do it with induction. And in fact, you could say that where we are now is we've localized the light switch on the wall. We found it. But the problem is that there are a whole set of switches, and we have to turn them on in just the right order. We're close enough now that we can usually turn on and turn off H-TERT the way your cells normally do, actually, in certain stem cell populations. The estimate is 6 months. I don't know if it's realistic or not. But here are the things we can test. Dermal one. So dermal studies in vivo in human beings currently planned for the end of next year, beginning of the year after. Current plan is to use transfection, using a gene gun, for example, and we'll come back to that. Technically, we're capable of doing the immune system now, but there's sort of a catch-22 in terms of who you pick for a patient population. We can do joints, but again, it's probably going to take a little longer. We could take chondrocytes now, take them out, and ex vivo put back the ones that we've transfected, but we haven't tried it. We could do arteries. It's tougher. It takes things like a double balloon catheter to do it. And we could probably try CNS, but it's a lot tougher in my judgment. With regard to the dermal approach, these are the patients we're going to look at. The first two, the dyskeratosis congenita and Fanconi's anemia, these are both patients that have demonstrated abnormalities in the way that they manage their telomeres. Right? It's complex, but the question is if we go in there and, in a sense, put in a normal gene and bypass the problem, which works in vitro, will it in fact make normal skin for these patients? The third one there, the Hutchinson-Gilford children, and there the problem is I've got about 40 of them in the world, and some of those aren't real Hutchinson-Gilford kids. They may be acrogyrics and metagyrics rather than progyrics. And if this was your child, would you let me try it on you? Would I let you try it on my daughter? I don't know. And a lot of these kids are in places we can't follow them anyway. And we need to do this in places with large medical centers. So we may have an N of 3. I'm not sure it's worth doing. But finally, and perhaps most importantly, we're going to be trying this on normal elderly people in wound care centers. trying this with things like venous stasis ulcers. That's what's in the works. If you want a little more about it, the first book there was something I put out now 6 years ago for the lay public. The second is just an urge. I want to urge all of you to submit to it. If we can put some more articles in Journal of Anti-Aging Medicine, I'd love to put them in. The third is the text that I have coming up. It's now excess of 4,000 references. It's 200,000 words. It would be excellent for propping up the corner of your couch. These are some other references. I reference one of my JAMA papers, only one of those, and a few others. And, you know, at this point in the lecture, it's sort of traditional to thank a number of people. If it was a lab, I'd be thanking all the people who worked in my lab. But I would like to thank, and so would the rest of the faculty, and so would the people who have underwritten this conference, and so would the moderators, you. Because without you, this wouldn't be a conference. Right. And furthermore, because you're here and your intelligence and your interest, we're able to have the conference this year and probably next year too. So again, thank you very much.
So, Your Highness, ladies and gentlemen, that now concludes our lecturers for the third Monte Carlo Anti-Aging Conference. But before we adjourn for the day, may I take a moment of your time to elaborate on why I believe we need anti-aging medicine? Let us overview some of the ideas that have been presented here in the last 2 days and study some of the changing demographics that are taking place in the Western populations. It's well known and accepted that life expectancy is getting longer. The average is increasing year on year. This particular graph illustrates the life expectancy for the United States from 1900 to 2050. Perhaps surprisingly, at present, the US average life expectancy is 27th in the world rankings. If you're interested, the top 3 countries, if you're thinking of emigrating to them, are Japan, Australia, and France. At the moment, the average man of 65 in the Western world can expect to live to be about 80, and the average woman can expect to be about 82. This difference can be more clearly exacerbated when you realize that a child born in 1997 can expect to live 29 years longer than one born in 1900, representing a 60% increase. Yet this could all be a drop in the ocean compared to what we believe is already possible. As we've seen, today's knowledge about the human biological system and the technology that is already available will mean that many people alive today will be able to live well beyond 120 and perhaps even more. And I'm sure after Dr. Fossel's lecture you may consider even higher numbers. So meanwhile, not only is the average lifespan becoming much longer, thus increasing the numbers of elderly individuals in the population, but we are also about to see the impact of the baby boomers upon society. The baby boomers are, of course, those individuals born between the Second World War and the Korean War. As the graph highlights in green, the baby boomers are now the single largest section of the population, and they're beginning to move into their geriatric period. Because of their sheer numbers, the baby boomers have obviously impacted every single area of commerce and society of their generation. Now we can expect this same impact for their geriatric needs. Geriatric, of course, is the term used for those over the age of 65. I have to say I find it rather disturbing that pre-geriatric is a term used for those over 45. Statistically, what does this mean? Well, it means that the elderly population is growing at a far greater rate than any other part of our society. For example, the growth rate of those between the ages of 15 and 64 will expand at only 6%, and those under 15 years of age at about 5%. You'll notice I'm giving you the sources at the bottom. This particular one is the World Health Organization. But individuals between 65 to 84 years of age are expected to grow at a rate of up to 200% between 2000 and 2050. However, the single biggest increase is attributable to those over 85, with an expected increase of more than 400%. So let's put all this into perspective. In 1900, less than 1% of the world's population was over 65. Today, 13%, 1/3, of the world's population is 65 or over. By 2030, not that far away, it's estimated to be by 20%, and by 2050, nearer 30%. So the demographics of aging are changing, but according to recent studies, so are people's attitudes about aging. When middle-aged people were questioned about their fears of aging, 46% were concerned about a declining health. 38% were worried about a lack of money. 13% were concerned about their mental faculties. And 12% were concerned about their possible dependence on others. However, 44% of people over the age of 65 described their present period of life as their best. And today, more and more people believe that old age begins with a decline in their physical or mental ability rather than the arrival of a specific birthday. As it stands today, an aging population is going to cost a lot, both in terms of the suffering to the aged individual and, of course, those effects they have upon their loved ones. Unless something can be presented to help alleviate this situation, these burdens will only increase within society. Consider that old age dependency rates will rise in every major world region over the next 25 years. The burden in 2025 is expected to be at least 50% larger than it was in 1998. In just 18 years' time, society will consider 1 in 6 of the population as elderly by current standards. But an aging populace could have benefits. On the upside, an increase of just 6 years extra life expectancy in the United States between the years of 1970 and 1990 translated into an additional gross national product of $57 trillion. And those figures were based on a 1992 report. But of course, treating age-related diseases and disorders comes at a great cost too. In June 2000, the University of Chicago estimated that curing heart disease would be worth $48 trillion, and that curing cancer would be worth $47 trillion. If we just look closely at one example of an aging disease, we can see more clearly the financial costs involved. For example, are you aware that Alzheimer's disease is currently the 12th leading cause of death? Alzheimer's disease is perhaps a good example of an aging disease because it rarely affects anyone under the age of 60. At present, there is a 1 in 7 chance of being diagnosed with Alzheimer's disease if you're over the age of 65. That figure rises to a massive and rather alarming 1 in 2 chance if you are over the age of 85. In fact, the risk of Alzheimer's disease doubles every 5 years after the age of 65. In the United States alone, $80 to $100 billion is spent every year on Alzheimer's healthcare or lost in earnings. It has been estimated that curing Alzheimer's disease would be worth as much as $1.5 trillion. So, if we take into account all the age-related diseases and disorders, is it possible to get an overall view of the financial costs involved? Well, let us first consider that all diseases fall into one of 4 categories. They are either inherited or genetic disease, infectious disease, trauma, or the degenerative diseases caused by aging. So, how much does each of these diseases account for in the fiscal budget? Well, looking at the United States Healthcare and Finance Administration figures of 1996 tells us this. Only 10% of all healthcare costs are spent on inherited, infectious, and trauma. In other words, 90% of the total healthcare budget is spent on degenerative disease caused by aging. 90%. And people say, can we afford anti-aging medicine? For the United States alone, that currently translates into $700 billion a year. So if we really want to make a big impact on healthcare in the world, we must focus on this 90%. We must focus on the degenerative diseases of aging. Because if we can slow or prevent the signs of aging from occurring, we could eliminate overnight the majority of disease and therefore the majority of its cost, both in financial and human terms. The world has to ask itself some basic questions. Can we continue as we are? Can we allow an ever-increasing elderly population to meet an already stretched healthcare budget? I'm sure all our newspapers, I know we're from 26 different countries here, I'm sure all our newspapers are full every day of how much it costs for healthcare and we can't afford it now. So can we allow, simply increase the numbers of people who will end their lives in suffering and misery? Should we expect the future working generations to pay 70%, 80%, 90%, 98% in taxes to support a full healthcare program for a top-heavy society? This becomes very apparent when it is estimated that if the policy of early retirement continues, i.e. between the age of 55 and 60, that in just 20 years' time, there will only be 2 people in employment for every retired person. It appears that older people will be a necessary component of the workplace. For one thing, there won't be enough young people around to fill the jobs anyway. But also, to be able to continue to contribute to society, if only in order to support themselves. I put this one in. Could we? I'll let you answer that question. Or will we introduce anti-aging medicine and revolutionize healthcare by concentrating on the true prevention of disease, thus drastically reducing the overall cost of health, adopting the use of natural substances and allowing our elderly to live in dignity and self-reliance, by remaining lucid and useful to the community at large? I believe to this challenge that anti-aging medicine arrives as the new logical healthcare. It offers a solution to alleviate many of the burdens of the burgeoning older population. Anti-aging medicine is the ultimate preventative medicine, and surely the next healthcare model for the new millennium. This model is based upon the very early detection, prevention, and reversal of aging-related disorders. We have the ability to alter the dreadful course of deterioration by inhibiting, delaying, and reverses the diseases associated with aging. In the United States, both the National Institute on Aging and the American Association of Retired Persons have recently recognized the significance of anti-aging medicine as the leading frontier of healthcare in the 21st century, and the science of anti-aging medicine is truly multidisciplinary. This significance can also be seen in the American Academy of Anti-Aging Medicine, as their membership of medical health professionals has risen from under 500 in 1993 to more than 10,000 last year. In these regards, Europe has a lot of catching up to do. As we've seen, anti-aging medicine is represented by advances in the field of biochemistry, biology, physiology, and enhanced by contributions from mind and body medicine, molecular genetics, and the new emerging medical technologies. The information being used by anti-aging physicians is also based on a global perspective, with its foundations in international scientific and clinical trials. This is because since the development of the internet and the coming of the information age, most of us are no longer prepared to operate within the confines of national boundaries and to restrict ourselves to what may or may not be approved, or indeed what may be approved, but only for some specific purpose. Anti-aging medicine accepts that aging diseases and disorders can and should be prevented rather than simply treated. Today there are literally thousands of physicians, scientists and researchers around the world involved in the research and treatment of aging. Indeed, we have met and listened to many of these leading lights here at the third Monte Carlo Anti-Aging Conference. We stand at the threshold of a new paradigm. For the first time in history, we're beginning to understand the principles of why we age, how we can measure biological aging, and how we can slow and treat its effects. Anti-aging medicine is fundamental. Through the use of nutrition, hormones, and some drugs, we now have the ability to delay, reduce, and even prevent the appearance of serious diseases such as Alzheimer's, arthritis, cataract, chronic fatigue, diabetes, and many other debilitating disorders. I envisage in the decade to come that thousands of anti-aging clinics will be established. People will attend these clinics for regular checks, and through the use of biological aging markers, an individual's rate of aging and risk from a particular degenerative disease will be measured. Steps will then be taken to slow and eradicate these biological aging signs before they become apparent, as well as difficult and expensive to treat. In other words, the traditional recognition and diagnosis of disease will change forever. One could consider these aging but otherwise healthy individuals attending anti-aging clinics in much the same way as individuals visit the dentist today for their preventative checks and measures. I can't talk about other countries, but 10 or 15 years ago in the UK, hardly anybody went to a dentist until they had a problem. Oh, I have a pain, I must go. Now, most people recognize the fact that they have to go not to have the pain in the first place. And I think it's a simple, logical step that we do the same thing in anti-aging medicine. The Romanian physician, Professor Ana Aslan, who many believe was the forerunner to the modern inception of anti-aging medicine, made this statement back in the early 50s, and I believe it's still true today. She said that successful aging is at the same time both a science and an art. However, as is usual with all great advances, mankind will probably experience the vested interests of the establishment and dogma that together will attempt to slow down or perhaps even try to prevent the wide-scale use of anti-aging medicine. We should remember the words of another very famous man who said, "Human progress has never been achieved with unanimous consent. Those who are enlightened first are compelled to pursue the light in spite of others. That man was Christopher Columbus after he discovered America. Today, a few insightful individuals realize that aging itself can be described as a disease, and indeed considered as such. But because everybody is suffering from it, we think it's normal and we accept it. Perhaps it's like asking a fish if it can see water, or a bird if it can see air. We too may not be able to see the wood for the trees. But once we grasp this fundamental fact and start to be proactive about aging, instead of just being tolerant, mankind will be able to take into its hands the possibility to radically alter its own path and its social structures. I believe that such a choice could expand our knowledge, understanding, and care of our world, leading on to new heights of intelligentsia and peace such as we've never seen before. Huge strides are being made in the understanding and control of the aging process. We hope that you will want to stay abreast of these fascinating developments here in Monte Carlo, and that you will continue to support us in making this anti-aging conference an annual event. That now concludes my official role at the Congress, and I wish one and all a very good afternoon.